Hello everyone and welcome to the first lecture in Bio 100. This is going to be the normal way that I give my lectures online. Please feel free to pause at any point, and if you ever have any questions, don't hesitate to email or comment or anything like that involving the video. I'm happy you could join me for this lecture and I hope you enjoy it. All right, so our learning objectives for this first chapter are pretty basic. We're going to be able to define science, biology, scientific thinking, as well as key aspects of well-designed experiments. That's going to be the scientific method. Spoiler alert there. We are also going to be able to describe how scientific thinking can be used to help make wise decisions, as well as the major themes in biology. So what is science and what is biology? This is a very broad question that we are going to be answering pretty much throughout the whole semester. In a way, this is a method for how we observe how the world works. You unknowingly use science every day, whether that is by asking a question or making an observation. Sometimes you might even be making a hypothesis without even knowing you're doing it. So a few questions that we have here that are some examples of what you might observe in the world and question about it. And this is kind of apropos to the time we're in right now, but are antibacterial hand soaps better than regular soap? How efficient are those hand sanitizers that smell really, really alcoholic, right? Why is morning breath so smelly, and can you do anything to prevent it? There's so many questions that you may have about the world that are based in science and also based in biology. And we're going to kind of talk about how you are a scientist. You didn't know it maybe until this moment, but you are a scientist. If you've ever questioned anything in the world, you've used science, and that makes you a scientist. Congratulations. So what is science? It's not just a body of knowledge. It is not just a list of facts to memorize. It is an intellectual process. It encompasses a few various things, such as observation, description, experimentation, that's a big one, and explanation of natural phenomena. So this is sort of your ability to observe the world around you, be able to describe it, pose experiments based on those observations, and then be able to explain those phenomena that occur. And last but not least, it is also a pathway by which we can come to discover and better understand our world. That is the root of science right there, being a pathway by which we can come to discover and better understand the world. So how do we know what we are seeing is true? If we move into kind of those observations that we see in the world, we can look at the various things around us that we are told by others or shown by others, and either we can take it at face value and just say, I trust you implicitly, you probably know more than me about this, or we can question what it is we are being told, or what we are trying to be sold. So can we trust the packaging claims that companies make? That's a big one. So that's what we're going to kind of talk about here for a second. An example of some claims include Cocoa Krispies stating that it helps with your child's immunity. I don't know if you guys know what Cocoa Krispies are, but it's just puffed rice with some flavoring, and we can kind of gather that maybe that claim that Cocoa Krispies cereal helps with immunity is maybe not very true. We can kind of look at that and immediately take it at that face value and say, I don't know if you're right there. However, you can look at another product, such as Airborne. This is going to be an immune support supplement that their big claim is help support your immune system. You'll notice that on this picture here, it has after that a little asterisk. Very, very sneaky way of packaging there. And I'm not knocking either of these products. I love Cocoa Krispies as much as anyone, and I'm sure Airborne does help maybe in some capacity with that little bit of vitamin C. But making these big claims makes us want to question whether or not they're actually true. So that leads us into that scientific thinking that helps put those claims to the test. And if we did do that, you'd find out that both of these claims are actually lies. Both of these products were approached by the FDA, which said, okay, give us your data. What did you do to get those big claims that you say? What makes that true? And both of them tried to put forward some bogus experiments that they did, and it turned out that it was all lies. So you kind of got to do a little bit more research. Sometimes you can tell right off the bat, I don't think that's true. Other times it's a little bit harder and you got to dig a little bit deeper. But that's where, the scientific method and our scientific thinking comes in. All right, so what is biology? Biology, if we break it down, are two words, bio and ology, which in the end lead us to the study of living things. Bio means life, and ology will always mean the study of. So if you ever see anything that ends in ology, it just means it's a study of something. So whatever that first word is, it's going to be the study of that. You can think like in oncology, or optometry is kind of a little bit of a weird one, but it's the same thing. It's basically saying that it's the study of something. In biology, we're going to ask very important questions, such as what is the chemical and physical basis for life and maintenance? How do organisms use genetic information to build themselves to reproduce? We will talk about genetics here in a few of our chapters coming up here. What are the diverse forms that life on Earth take, and how has that diversity risen? How do organisms interact with each other, with their environment? We are actually going to talk about almost everything on this list here. So those are some important questions that we need to ask when it comes to biology, the study of living things. Moving on to scientific literacy. So this is how we begin to think scientifically. We are going to use the knowledge that we gain from what we observe, what we research, what we figure out for ourselves to make wise decisions. For example, whether or not you want to purchase the immunity boosting Rice Krispie treats for your kids. Scientific literacy is incredibly important in all of our lives, and you do use it daily. There's a great chance that something probably stuck in one of your science classes, and you'll remember it every once in a while. And hopefully from this class you'll find more things that you remember than you forget, hopefully. It is especially essential in matters of biology, not just in day-to-day life, but especially in biology. And it's also going to help you with a nice balance of healthy skepticism. Now, healthy is in quotes because obviously you should be skeptical of a lot of things, but you shouldn't be paranoid, right? You don't want to be paranoid about every little thing that you purchase or that you do. That is not going to lead to a happy life, but it is healthy to question a lot of things, but to a certain degree. All right, so biological literacy is essential in the modern world, and it is honestly everywhere you look. So a lot of these examples that I have here for what you see when you think of when you should think of biological literacy in our modern world is climate change. That's a big thing going on right now. One of the products that I think is really, really cool, some people have done it, maybe you have, but it's that 23andMe or some type of company that runs your DNA and tells you about your ancestry. There's a lot of things revolving around agriculture right now, GMO products, allergies, whether or not your diet is healthy. So that involves like saturated fats and unsaturated fats, those various things. But you're going to see a lot of that everywhere you look. And you might not think of it necessarily as science or biological literacy, but it is. And your ability to kind of take in that information and understand it in some capacity, or whether you care about it at all, that plays a big part in all of this. So through this course, I do hope one of the goals is that you develop your biological literacy a little bit more than it currently is, so that you become a little bit more aware of your understanding or shortcomings of what's going on around you in the world and how much you should maybe care about those things, I guess. Okay, so your biological literacy is the ability to, 1, use the process of scientific inquiry to think creatively about real-world issues, and 2, communicate those thoughts to others, as well as integrate them into your decision making. So that is the hardest part of biological literacy. Using all of these things, it is important. So being able to process, communicate, and then integrate all of these facets into your day-to-day life. And it's something that you kind of have to practice at a little bit. But hopefully through this class you can kind of learn how to balance all that. Okay, so moving into thinking like a scientist, how do we use the scientific method? Now, the scientific method is probably one of my favorite things to teach students or people in general when talking about science, because it is such a cool and evolving process that really you don't think about until you have to think about it. But you've done it. There's a great chance that you have already done this process. It does have a tendency to change the order of things that you do. As far as this course goes, this is just the in general order that we are going to put the scientific method, but this can change a little bit. So maybe you learned it one way, maybe you see it a different way if you look it up, but this is kind of just in general for this course. So scientific thinking is going to be empirical. Scientific thinking is based on, and make sure you know this, experience and observations that are rational. This is very important, testable, and even more important, repeatable. If you can't test it, and what is tested cannot be repeated, it is not good science. Okay? That is very important to understand. Scientific thinking is also self-correcting. It is an advantage to be able to change your mind if it turns out you're wrong. That is one of the best things about science and obtaining data. If you're wrong, that's okay. You don't have to be right. I've always been told, and I tell students the same thing, no data or wrong data is still data. And that's important to understand. Even if you're wrong with what you were guessing would happen, and it turns out you were really incorrect, that is still data. And that's still important. As long as you can test it and repeat it, then you're good. You're good. Okay, so jumping into this scientific method, the first step is to make an observation. Typically, you're going to do this with one of your senses. So scientific thinking begins by making observations about the world. For the example that we are looking at, the observation that we are going to be looking at is that plant roots appear to always grow down into the ground. So we're going to kind of start to question how a seed knows which way is down instead of growing up, right? So we are making a little observation here about plants. Our second step is to formulate a hypothesis. Now, I don't want you to get scared if you have a traumatic experience from grade school or middle school or high school or whatever about making a hypothesis. This is not going to be a scary process, I promise. I think hypotheses can be daunting to write, but don't be scared. So a useful hypothesis is going to be one of two things. Well, they're going to be both things. It must clearly establish alternative explanations for the phenomena occurring. And the second thing is it must generate testable predictions. So a hypothesis, make a little note, is not a prediction. Okay, it is not a prediction. We will talk about what a prediction is later, and that's going to look more like an if, then, because. So hypotheses are never, usually, unless it's taught to you incorrectly, stated as an if, then statement. Okay, the hypothesis that we're going to kind of continue with our little plants that we're observing the roots is that seeds know which way is down due to gravity. That is the hypothesis that we are making, that seeds know which way is down due to gravity. As we talk about hypotheses, we need to talk about two different types of hypotheses, a null and an alternative hypothesis. Okay, the alternative hypothesis, we're going to give you an example here, is that gravity does affect direction of root growth. So this is an alternative hypothesis. A null hypothesis is going to be the negation of the alternative hypothesis. Gravity does not affect the direction of root growth. So make a little note, an alternative hypothesis is usually kind of like, if you want to think of it this way, a positive. A null hypothesis is the negation or negative of what the alternative hypothesis is. So moving into a little bit more about null hypotheses. A negative statement proposing that no relationship exists between the two factors, so just like we said, adding that negative not or no, does not, etc., is going to make that into a null hypothesis. These hypotheses are equally valid, but they are easier to disprove. So you're going to see a lot of null hypotheses through scientific data, honestly, because they are easier to disprove. That's honestly something you kind of want when it comes to creating a scientific paper that is a valid paper. It is impossible to prove that a hypothesis is absolutely and permanently true. That is one of the most important things I want you to understand about anything we talk about, is that it is absolutely impossible to prove that something is completely, absolutely, infallibly true, okay? It doesn't matter if it really seems that that is the only option, okay? It is important to understand that science is constantly changing, evolving, getting better, and looking at various problems or hypotheses that have been formed in the past, taking that information and making it better and making those statements stronger. And that's fantastic. But there's very few scientists, I'd have to say, or biologists or anybody in the science community that will absolutely say, beyond a shadow of a doubt, you know, hand on the Bible, etc., this is completely true and there's nothing that's going to change it, because things change all the time. New data gets created or found, new information is discovered, and things change. It's the beauty of science. So asking questions and proposing hypotheses. Some scientific questions are causal. Now, please read that carefully. And that says causal, not casual. It's not asking, you know, about the weather and, you know, how your kids are doing. It's a causal question, like cause and effect. These lead to hypotheses about what is responsible for observed phenomena. An example, does gravity play a role in seed cells knowing which direction is up? That is a causal question. Gravity is affecting the direction of of seed and root growth. Causal, cause and effect. Some scientific questions are going to be descriptive, but those are not necessarily the ones that we want to focus on when creating a hypothesis. So they can lead to interesting discoveries, which is fantastic. We want that. That's exciting. But it's not going to be answerable by a hypothesis. Okay, for an example, what is the range of a bald eagle? That is not going to lead us to a hypothesis that can be answered. Okay, jumping into step 3, we're going to devise a testable prediction. Our goal here is to propose a situation that will give a particular outcome if your hypothesis is supported, but a different outcome if your understanding is not supported. Okay, so remember when I said we're posing a hypothesis versus a prediction, predictions are if-then. If this happens, then this will happen. For example, in our little experiment that we are conducting in this example, if gravity affects the direction roots grow, then we should see atypical root growth in microgravity. Meaning that if gravity really does determine the direction that our roots grow, if gravity does cause roots to grow down, then we should see our root growth do something weird, something kind of funky and out of the normal if they are in microgravity, which is going to be different than our normal gravity, right? So if then, that is our prediction based on our hypothesis and our observation. All right, so step 4, we're going to conduct a critical experiment. This makes it possible to decisively determine whether a particular hypothesis is better than an alternative hypothesis. So these experiments are going to be put together. We're going to form some type of idea of how we're going to test our hypothesis, and wonderful things happen sometimes, and sometimes they really don't. Once we have some type of testable situation going on, we enter into step 5, drawing conclusions from our results. So if step 4 goes well, which I'll be honest with you, it normally doesn't. You have to go back and do it again and do it again until you figure out how your experiment needs to be run. Sometimes you run into technical problems, sometimes you run into logistical problems, it just depends. But once you get something that works, you're going to get results. You're going to get data. And from that data, we're going to draw conclusions and make revisions, whether that's to go back and redo something, to keep testing, it just depends. We're going to be looking for patterns and relationships in our evidence. We're going to determine if findings and conclusions support our hypothesis. If they do, cool. If they don't, still cool. If the result is not what you expected, that doesn't make it wrong, doesn't make it a wrong answer. You might need to adjust your hypothesis, which happens frequently. Remember, nothing is set in stone when we are doing this process. It is part of the process to adjust and make reparations to what you're doing. From that point, you may have to conduct additional experiments or redo the whole thing. Sometimes, and it really isn't fun, you have to scrap the whole thing and just redo it. So when do hypotheses become theories? And what the heck are theories anyway? So a hypothesis is a proposed explanation for a phenomenon. A theory is kind of like a hypothesis, but there's a very big difference. A hypothesis is going to lead to, make sure you underline it, testable predictions. Theories are not speculation or guesses. They are broad hypotheses of which scientists are really certain that it's the right answer. They are tested, but they can't necessarily be tested. They are a broader scope than a hypothesis. They're very broad. One of the things about theories is that they're hard to test. We try to test them. Sometimes they're hard to test. A few examples of some theories include gravity, evolution, cells, germ, plate tectonic, game theory, special relativity, general relativity, quantum theory, and heliocentrism. So some of these that you probably are familiar with are the ones I'm going to kind of talk about very briefly. Gravity and evolution. We can't 100% say beyond a shadow of a doubt that gravity exists. I mean, we know it does, right? It's a highly supported theory, okay? Gravity is highly supported. So is evolution. Whether or not you believe in it, it is still very, very supported by evidence. All of this is supported by evidence, but we have a hard time testing it. You can't necessarily throw animals or plants or whatever into a chamber and watch evolution. It takes too much time. Likewise, gravity isn't necessarily something that we can just see. I mean, obviously you can watch something roll off a table and fall, but you can't 100% say it's gravity, if that makes sense. I know that's confusing, but theories are like hypotheses. They're just really, really broad, as well as hard to test. All right, so there are a few terms that we need to kind of define and also talk about. So the first is going to be treatment, any experimental condition applied to a group. So this is whatever you're doing to collect the data, what you are performing onto the subject of your experiment. You have your experimental group. This is a group exposed to a particular treatment. So the group that is going to get the first part of this, the treatment. Your third group is your control group. It's a group of individuals who are treated normally and identically to the experimental group, with one exception. They're not exposed to the treatment. This is important to include. So if you are testing, let's say, bone density, birth control's effect on bone density, you are going to want to have your experimental group be taking the birth control. You are also going to need a control group of people who are not taking the birth control, so that you can see if there actually is a difference between taking the birth control and not taking the birth control on bone density. A few variables that we need to discuss, which is always everyone's least favorite thing when it comes to talking about scientific experiments, is going to be your independent and dependent variables. These are going to be characteristics of your experimental system that are subject to change. We'll talk about those next. So these controlling variables, we're going to talk about these first. I lied to you on the last slide. The most important feature of a good experiment are the controlling variables. The attempt to minimize any differences between a control group and experimental group other than the treatment itself. This is incredibly important when supporting your data and making the claims that you are making about your experiment. When there are no controls, so poor experimental design can lead to flawed conclusions. Without a control group, we can't really support our data. Okay, these are going to lead to flawed experimental design with no controls. So in this instance, we are looking at 24 individuals that have gastric ulcers, and all 24 of them are going to receive the experimental treatment. There are 0 individuals in our group that are part of a control treatment, so they're not receiving any treatment. We are going to fill a balloon with refrigerated fluids and it is placed in the stomach. So that is the treatment that we are having to treat these gastric ulcers, which are basically sores inside of the stomach. The results will show that 100% of patients experienced relief. So because all subjects experienced relief, it's tempting to conclude that the treatment worked. Now, if we include a control group, so we've got 160 individuals with gastric ulcers, everybody's got an ulcer, 82 received the experimental treatment, that balloon filled with refrigerated fluid, and then 78, our control group, received a balloon with room temperature fluid, and that's placed in the stomach. The results that we have here are that 34% of patients experienced relief and 66 didn't. 38% of our control group experienced relief from that just room temperature balloon, so that control, and 62 patients experienced no relief. So compared with proper control, gastric freezing didn't actually result in any benefits. So you see why a control group is very important to actually support the data that you are claiming. You have to have that. Otherwise, there's a good chance that your experiment is actually really flawed and not good. You've probably heard of this before, the placebo effect. This is a phenomenon in which people respond favorably to any treatment. Okay, as long as they are receiving something, it's sort of this mental state that they are receiving something good. Mind you, the placebo effect only really works if the person doesn't know that they're not actually receiving the treatment that they think they're getting. So if you give someone, let's say a sugar tablet, but you tell them that it's for pain relief, they have a headache, let's say, and you give them a little tablet. When they take that tablet, they might feel relief in their headache, but in reality, you know that they don't know that that shouldn't have worked, but they sort of think themselves into feeling better from that treatment, even though physiologically it has no basis for making that person feel better. So the placebo effect highlights the need for comparison of treatment effects with an appropriate control group. Okay? Something kind of interesting about doing experiments is something called bias, as well as influencing an experiment's outcome. A wonderful example is this lovely horse. His name was Clever Hans. Hans only knew how to solve a problem, mathematical equations, if the person asking knew the answer, and Hans was able to see them. If Hans, the horse, could not see the person asking the question, then Hans didn't know the answer. Likewise, if Hans's questioner didn't know the answer to the mathematical equation, Hans also didn't know the answer to the mathematical equation. What was discovered later was that Clever Hans maybe wasn't quite so clever. He was just really good at reading body language. So if he was getting close to the answer, something changed in the questioner's body or face. Something happened, and the horse was able to read that and know that he hit the answer. And by the way, he would just sort of stomp his hoof however many times to say the number of the equation. So if you asked him, you know, hey Hans, what's 4 plus 4? He would stomp his hoof 8 times. Of course, he'd have to see you, and you'd have to know the answer for him to get the right answer. So that is influencing an experiment's outcome. The horse actually didn't know the answer. He was just able to tell what he was supposed to answer by the experimenter or the asker of the question. So designing experiments, you have a few things that you need to kind of keep in mind. A blind experimental design, it's the experimental subjects do not know which treatment they're receiving, if they're receiving a treatment at all. Again, this is sort of kind of goes hand in hand with a little bit of that placebo effect that I was talking about. Giving someone sugar tablets instead of giving them painkillers. But these subjects won't know that they're receiving sugar tablets or the actual treatment. A double-blind experiment is going to be where neither the experimental subjects nor the person performing the experiment, the scientist, nobody knows which treatment the subjects are receiving. So you don't know you're giving your subject the sugar tablet. You just know you're giving them something. That's it. You don't know what it is. You wouldn't know until the end. And then last is randomized. This is where the experimental subjects do not know which treatment they are receiving. All right, moving into biases. This is also kind of a way that can influence the result of an experiment. The percentage of papers published with female first authors is what we are looking at. We are seeing data on the left when the reviewers knew the sex of the author. And then on the right, we see when the reviewers did not know the sex of the author. And this is all kind of dependent on what papers were being published. So when the sex of the leading author was known by the reviewers, fewer papers by women were accepted for publication than when the reviewers did not know the sex of the author. So, interesting stuff. You can't be biased. Repeating experiments is going to be really important. I know I discussed this before, but replication is the process of repeating a study. It's going to increase our confidence in results, so whether or not we really support it or we don't. And it's going to help isolate variables responsible for outcomes of experiments. Experiments that can be repeated again and again are an effective defense against biases. If you perform an experiment, let's say, and you come to a conclusion based on your data that you tested and you feel pretty good about, and then your paper and your experiment are up for peer review. If one of your peers in your field is able to repeat that test that you did, your entire experiment, and come to the same or a similar conclusion, that is awesome. And that is the point of peer review. If your experiment cannot be replicated, there's a good chance there was some bias in there somewhere. So here we have a visual display of data that can help us understand and explain phenomena, various types of displays, a bar graph, a line graph, and a pie chart. All of them are used in various ways, and there are right and wrong ways to use these displays. This can go in kind of like hand in hand with biases. If you want to try to make your paper look like it's saying one thing, you might use a line graph instead of a bar graph or a pie chart. It may change how the reader is going to interpret your paper. So again, these are common elements of visual displays. You're going to have a title. This is very important to have. One, it tells you what you're looking at, and you need that. Your data points are going to go throughout the display. It's very important to put your data in there. And then the things around the data. You have your y-axis and your x-axis. The x-axis is going to go along the bottom while the y-axis goes along the top. It's going to be that vertical axis versus the horizontal axis. You need to have both of those axes labeled. That is going to be your independent variable on the x-axis and the dependent variable along the y-axis. This should have some type of title of what is going along that axis, as well as what is being represented. So in this case, along the x-axis, we have hours, and then along the y-axis, we have a percentage. So you should always kind of include what's going on there and what you're quantifying. Okay, finally, we're talking about variables again. I do apologize. I thought that was the next slide. Teachers, we're not always right, are we? All right, so variables. We have our independent and dependent variables. Let's talk about them a little bit. Our independent variables are going to be where some are measurable entities that is available at the start of a process and whose value can be changed as required. And these are generally represented on the x-axis, as we discussed on that last slide. Our dependent variables are created by the process being observed and whose value cannot be controlled. So make sure that you know the difference between independent and dependent variables. Okay, kind of jumping back and forth between what you want to see and what's not good when it comes to data, back into misleading displays of data. So how can a graph be misleading? It can be ambiguous in labeling or scales. You might make your data points further apart than they actually are to try to show a bigger difference than what's actually there. And incomplete information on how data was collected. This is a great way to skew your data and make it harder to repeat. Biases or hidden assumptions, and these are not always done on purpose. A lot of times they are done in a not so good way, but when that happens, the science community has a tendency to sort of band together and say, not in our house. Unknown or unreliable data sources. Sometimes if someone wants to get a point across in their paper, even if it's not completely correct and potentially not good of them to do, they'll try to reference papers that don't actually exist or reference sources that aren't real. And it sounds kind of weird, but essentially think of it like cheating. It's just a bunch of cheating. And then the last is insufficient or inappropriate context for data presentation. So using the wrong type of chart or graph to display the data. That, again, makes it to where the reader looks at it and says, okay, I get it, it's that one thing. In reality, they displayed it in a way that makes you think it is that way when it's really not. Moving on to one of my least favorite things that must go into papers is statistics. Now, I say that, but they are one of the most important things that are included in journals and articles and papers. You have to have statistics. It is a way of running your data, taking your data and turning it into an equation to get a result and be able to draw conclusions from the data and actually be able to present it. You have to quantify your data and your results. It is absolutely the most important thing you can do. If you don't do this, your paper basically doesn't matter. You have to do these things. So these are statistics are a set of analytical and mathematical tools. So typically these are tests, not ones that you actually do like an experiment on, but equations that you run. So these mathematical tools are equations that you run, designed to help researchers gain understanding from the data that they gather. Do keep in mind that drawing conclusions based on limited observations is very risky. If you have a really small amount of data or a small amount of subjects in your data, this can result in not so good statistics. So if you are trying to say that, let's say, vaccines cause autism, and you only use 12 children in your whole experiment, and then you make this broad statement that vaccines cause autism, you're going to be looked at really funny. What is very sad, though, is that that paper was published, and it got circulated, and it got some ground, which is very unfortunate because the man who ran that experiment, Wakefield, only used 12 subjects, 12 children, and he made that big claim. Now, if he had used, I don't know, 100, 500, something like that, a larger amount of subjects, maybe we could look into that, but he didn't. And he also had a lot of bias because he was paid off. So sorry if that burst a bubble for you, but that guy was very wrong and is honestly probably one of the banes of the scientific community. His paper was pulled from the article. It was completely discredited and could not be repeated. No one could repeat the data that he got. And that tells us that his experiment was wrong, that he was biased and he was influencing the data, and it was exclusively for money. So, not good. I'll get off my soapbox. So, drawing conclusions based on our statistics. So once we have our statistics run, after we've received all of our data and all that good stuff, we are going to draw some conclusions. The greater the difference and the smaller variation between the two groups, the more confident we can be that our outcome did not occur by chance. Now, that kind of sounds a little bit weird, but essentially it just means that we can be really confident that our data is good, that our statistics are good, and that our results are good. Okay, so here we are going to talk about how statistics can also help us to identify relationships or the lack of relationships between variables. There's a few statements I want you to know. A positive correlation, meaning that when one variable increases, so does the other. That is a positive correlation. Likewise, there is an opposite, which is a negative correlation. As something decreases, so does something else. Correlation is not causation. Put a little asterisk or star next to this because that's very important. Just because that correlation occurs, when something increases, so does the other. When something decreases, so does the other. Just because that occurs does not mean that it is the cause of that process occurring. And statistical analysis help us organize and summarize observations and experimental evidence. So again, going back into kind of how things can be very misleading. So misleading evidence can obscure the truth. This is something we can call pseudoscience, where individuals make scientific sounding claims that are not supported by trustworthy, methodical scientific studies. So phrases like 4 out of 5 dentists recommend using Sensodyne, or whatever it is that you're trying to be sold. So it makes it sound very scientific and supported and trustworthy. Always do your research. It's important. The second is anecdotal observations. This is based on only one or a few observations. People conclude that there is, or not, a link between two things. And this is something that's basically like superstitions. So here we have an example of vitamin water, where they're using revive and essential. So these scientific sounding words are there to try to convince you that vitamin water is essential for your revival, etc., etc. They're keywords that are meant to play on your desire to be healthy or your desire for something. There's a lot of makeup products where it talks about hydration, or it helps with aging and things like that. You have to do your research to see what ingredients are included in these various things that actually support what they're saying. Does it actually revive anything? Is it essential for you? I've lived my whole life not drinking vitamin water. Am I missing something essential? Probably not. So bad science leads to dangerous behavior. I already talked about Wakefield and his paper claiming that vaccines cause autism. The short answer of that is an emphatic no. There is nothing that supports that vaccines cause autism or any other type of health problems. Truly, sincerely, I swear to you that it is a hype meant to scare you away from taking vaccines. Whatever it is that is trying to convince you of that, a holistic approach, someone claiming that they have something like, I don't know, a magical spirit that's going to cure you of what ails you. You know, they are doing it to benefit themselves instead of benefit you. I can't think of any scientist who came up with a vaccine who didn't have a reason that was personal to them, and then provided those vaccines for the public so that it would benefit the public. For a very long time, many diseases that were highly contagious became extinct, completely extinct, until a vaccine scare started occurring in our current times. Polio was a terrifying thing to have. Almost no one in modern day has polio because of the vaccine. There's a good chance that's going to occur with chickenpox. A lot of people in the most current generations receive a chickenpox vaccine, and very few people now have chickenpox. So that is just sort of the trend that occurs. So anyway, I'll get off my soapbox again, but that is something that's very important to think about in how bad science leads to dangerous behavior. Now, I don't tell you these things trying to make you question, well, is science really a good thing then if it can be influenced? Everything can be influenced, from religion to social medias to movies to music, video games, shoot, whether or not you have a particular type of shoe brand, toothpaste, everything can be influenced, and it can influence you. What I'm saying is, question it, try to figure out whether or not it's correct, and now you kind of have the tools to do that. You can kind of question these things and use your scientific literacy to do that. I'm not going to go through this again. I'm so sorry, guys. I really did jump on that high horse a little bit too soon. But just remember that you should question big, broad claims such as a link between vaccines and autism. Autism is caused by many various factors that can be biological, it can be emotional, it can be mental, it can be genetic. So there's a lot of things that can influence autism. Vaccines are not one of them. Now, I know I'm selling science like it's the coolest thing ever, because it really is. But there are limits to what science can do. The scientific method is above all empirical. Value judgments and subjective information fall outside of the realm of science. Science neither generates moral statements nor gives insight to ethical problems. So if it's something a little bit more on the emotional side, science can't really help with that. Okay? It is there to give you an answer about the world, but it is not going to give you an answer with feeling and emotion. Technology is not science. It is the application of research. That is kind of an important thing to make note of here. So what is life? It's an important theme that unifies and connects diverse topics in biology. But really, what is it? If you try to think about it and define it, what would you quantify as life? Is it at conception? Is it anything that lives, breathes? Is it things that grow? Is it things that we see around us every day? Is technology life because it evolves? Is it everything that can evolve? Is it things that can change? So try to define life in your own words. Think about it, write it down, I don't know, do whatever you want to do with it, but I want you to think about what is life. So I want you to have in mind the answer that you thought of for the last slide. And then I want you to go through this list with me. These are characteristics that have been determined by scientists that are shared by all living organisms and living systems. The first characteristic is that it is a complex and ordered organization consisting of one or more cells. So to be a living organism, it has to have one or more cells. The second thing is they use and transform energy to perform work. So for humans, that'd be like taking in water and food. For plants, that'd be like sunlight and water. The third character is sensitivity and responsiveness to the external environment around them. So being able to respond to what's going around you. Regulation and homeostasis. I like to classify homeostasis as being the optimal internal state. So it's basically just when you're alive and you're keeping yourself alive. So it's that process of everything working together to keep you living. The next character is growth, development, and reproduction. Reproduction is a big one, a big characteristic of a living organism. And the last is evolutionary adaptation leading to the descent with modification over time. So it's just basically saying that there is a change that occurs through time, basically evolution, which is just change. There are two unifying themes and four major areas of focus in biology. Those two themes are hierarchical organization and the power of evolution. The areas of focus include chemical, cellular, and energetic foundations of life, genetics, evolution, and behavior of individuals, diversity of life and the unity underlying it, ecology, the environment, and the subtle and important links between organisms and the world they inhabit. And from there, that concludes Chapter 1. I hope you enjoyed this. I hope it wasn't too boring, and I hope you will join me in the next chapter. Have a wonderful day and make good choices.